Hello? Hello, Soham.
Yes, sir, bolun.
Any question regarding the animal cell culture?
No, sir.
বা কোনো কোশ্চেন বা টপিক রিগার্ডিং এনি টপিক রিগার্ডিং মাইক্রোবায়োলজি that will be not covered বা do not understand সে ধরনের জন্য টপিক থাকে, না হলে আমরা কিছু অ্যাসেপটিক টেকনিক দেখি যেখানে সেল অ্যানিমাল সেল কালচারের জন্য কাজে লাগবে। at this time it will be the first step of the practical aseptic technique. Aseptic technique তোমাদের ইউজ করেছিল না, প্র্যাকটিক্যালে ছিল অ্যাসেপটিক কন্ডিশন। হ্যালো হ্যালো সোহম,
হ্যাঁ স্যার বলুন।
বলছি অ্যাসেপটিক কন্ডিশন তোমাদের ছিল তো প্র্যাকটিক্যালে?
ছিল ছিল ছিল
ছিল। তো সেটা প্র্যাকটিক্যালে দেখেছো দেখেছো বা দেখোনি জানি না, দেখেছো?
না
সেটা
এখনো।
অ্যানিমাল সেল কালচারের জন্য খুব ইম্পর্টেন্ট, মানে অ্যাসেপটিক কন্ডিশন। তো টুডে উই ডিসকাস অ্যাবাউট দ্য অ্যাসেপটিক কন্ডিশন এন্ড হাউ ইট উইল বি মেনটেন ইন কেস অফ অ্যানিমাল অ্যান্ড টিস্যু কালচার, সেটা আমরা দেখি। ঠিক আছে, ওকে?
ওকে স্যার।
যদি কোনো বা অন্য কোনো যদি টপিক থাকে বলো, আমি বেটার আই ডিসকাস উইথ ইউ।
না স্যার এটাই পড়ান। হ্যালো।
আচ্ছা, you can write in the name and roll number again please, কারণ যা সেটিং এ চেঞ্জ হয়ে গেছে। অনলি দা নীলাঞ্জন ঘড়াই আরেকবার লিখে দাও প্লিজ। হুম, okay. Hello, this is the animal cell culture basic. The very important is the to aseptic condition to maintain the aseptic chamber. You can just see what are you doing now for aseptic condition. So first of all you sterilize the aseptic chamber, then you wear the apron, a sterile apron. That's why they want separate room here. To before enter into the aseptic chamber, you remove your own wires, because wires and hands and hair, which is the main component for the contamination of the microorganisms. So be careful. Then the air, wash your hands. This is the one of the most important technique. Wash your hands. Hello. হ্যাঁ, আচ্ছা, তুই তো এমন সময় ফোন করলে, এখন তো ক্লাসে। বললাম, কিন্তু এখন তো ক্লাসে আছি আমি। তুমি একটু ডিটেলসটা পাঠিয়ে দাও আরেকবার, ঠিক আছে? মানে তোমার নাম, রোল নাম্বার, এই, ঠিক আছে? হ্যাঁ হ্যাঁ, ঠিক আছে। Hello, now the it will be washed or sterilized by the alcohol or by the hand rubs or sanitizers. Now all the media and others preparation will be sterilized before it is used. Then all this preparation will be kept in sterile condition, and whole room must be free from the microorganism.
Flow through the chamber, which will not maintain the sterile field. For the same reason, it is important to keep the front sash in the lower position when working. The basic idea is to keep your media and reagents sterile by only.
The basic requirement is the to keep sterilize the media and the reagent also.
Keeping your cultures free from contamination.
So for the free for the contamination, this is very important. Otherwise, it will be contaminated of the newly or growth of the cell and tissue. Next, we use some pipettes for the pipettes for adding some reagents or the cultures. So first will be by the gun, it will be cover it, and this discard it very carefully, otherwise it will be contaminated.
When holding the cap, it is important to touch the inside edge, or you could contaminate it. Replace the cap as soon as possible. If you must set the cap down in order to free your hand, set it down with the interior surface facing down. When pipetting, try not to touch the pipette to anything non-sterile, particularly the outside of containers, or contamination could result. Each pipette should be used one time and discarded, or washed if using glass pipettes. Reusing pipettes by dipping directly into another media bottle or by
Do not reuse the pipette. This is very important. Sometimes most of the students generally use the one pipette can be used for the every purpose, but this is not the true way, because one pipette used for the one reagent then add to the another reagent, that will be difficult, that will be contaminated. So be careful. Basically use for one separate pipette used for the separate reagent, and this is used for the culture media for the preparation. Before you cut the tissue or the animal tissue or the plant tissue for subculturing, so that's why they transfer the culture media first, then another pipette we used, and it will be used by the gun. Measurement volume of the solution will be kept out by the pipette. A very good
I'm a molecular researcher here at the University of Toronto. Harvest and treat so that you can see them for your lab purposes. In order to environment, because if things like mold or bacteria were to get into your samples, they would invalidate the results and you would need to start again. Airflow hoods work by generating a barrier between the external environment and the internal working environment. DMEM, or Delbecco's modified Eagle medium. This media provides salt balance, pH balance, and essential nutrients so that the cells can survive. We also supplement this media with 10% fetal bovine serum. Fetal bovine serum is the plasma from a fetal cow. And what we do is we harvest that because it contains a large number of growth factors that will then stimulate our breast cancer cell lines to grow. We also use a 1% prophylactic administration of penicillin and streptomycin. What that does is it prevents any potential bacterial contamination that may occur. In order to prep this bottle, we spray it using a 75% ethanol solution. That disinfects the surface of the bottle and it is now ready to be placed in the cell culture hood. The next reagent we are using is 1 times phosphate buffered saline. This is just a basic saline that is buffered to pH 7.3 that we can use to wash off any lingering remnants of media that may be on the cells. Again, it needs to be disinfected. Finally, the last reagent is 1 times Trypsin Edta. This is a digestive enzyme that digests the external cellular proteins so that the cells lift up from the bottom of the plate and are in suspension. From that, we can then dilute them and replate them in different plates. That is called passaging cells. Now that our reagents are warmed and disinfected, collected and we've placed them in the cell culture hood, we are ready to begin the process of cell culture. The MCF7 breast cancer cell lines are adherent cells, meaning that they adhere to the bottom of the culture vessel that they are being grown in. As you can see, there are multiple examples of culture vessels. This one here is a cell culture dish. The lid comes off, and there are multiple sizes that can be used depending on the nature of your experiment. This is a cell culture flask. Cell culture flasks, as you can see, are fully sealed and they have a lid. There are pros and cons to both the dish and the flask. Dishes are easier to work with, but there's a higher potential for contamination as the lid comes right off. Flasks are a little more awkward to work with, but they are protective of the cells because they have a lid. For the purposes of this video, we will be using the cell culture dish. This is a cell culture incubator. It maintains conditions of 37 degrees Celsius and 5% CO2, which are similar to the internal conditions of our bodies. Our cells are growing in a cell culture dish with inside. Let's take a look. As you can see, there are many cell types being cultured in this incubator. Our cells are the MCF7 cells. And now that we have them from the incubator, we can take them and passage them in the flow culture hood. Now we are ready to passage our MCF7 cells for our experiment. How we do this is we will remove the existing media, rinse the cells with PBS, and then place trypsin on the cells. The trypsin will digest the external proteins of the cells, allowing them to lift up off the bottom of the dish. Then we will dilute the cells in new media and place the newly diluted cells into new plates that contain new media so that the cells can now grow in different size plates for our experiment.
So same type of things is used for. First you collect the MCF7 breast cancer cell line, the phosphate buffer solution. Before the same technique, all the reagent before it is used, there may be sterilized by the 75% ethanolic solution. Just before you can see it, yeah, spray by the and another pipette is used for another pipette. This is contain the pepsin, the distinct enzymes. All the reagent is used by the separate pipette very carefully and in the very aseptic condition. Otherwise, it will be contaminated. This is the third pipette used for the use of trypsin for cell-free or digestive enzymes to do not adhere the cell. Every one gun is the vacuum pump is used. Don't use the mouth for suction of the pipette or volume.
Initially, after the cells have been passaged, they will be quite separated and will have a lot of space around them. The population density of cells in your plate is called cell confluence. Confluence is measured subjectively by comparing the amount of space surrounding the cells. If the cells cover 30% of the plate, then the plate is 30% confluent. If there is virtually no
There are
space
two
around the
plates.
cells, then
One is the
plate
30%, another is the 75%. So where the
confluence. Generally, when cells are too
growth
close
of the
together,
cell is much more higher in the second plate.
apoptotic pathways are then upregulated in the cells and they begin to die. In order to prevent this, we passage the cells before they reach 100% confluence. Now that we've passaged our cells, we put them back into the incubator and allow them.
Successful cell culture depends heavily on keeping
এটা থেকে
Systec, the autoclave company, is a leading manufacturer of steam sterilizers and media preparation systems.
So we can see the animal cell cultures, the types and the culture techniques and the application. These are the basic topics. The last 2 to 3 class we are discuss about doing the how to the animal cell culture or the tissue culture will be established. The tissue culture is the general form of the removal of the cell, tissue or the organ from the animals. And so basically the tissue is collected from the plant, an animal or others, and their subsequent placement into the artificial environment and suitable for the growth. This is the subculturing procedure, and the cell were the removal from the organ fragments and separated in the single cell condition and placed into the suitable culture media. This is very important because the all cells are not in the same culture media, so there is a different culture media. You first prepared or purchase it before you before you add the cell or tissue. That method is called the animal cell culture. Next, there are the basic steps to establish the primary cell culture. First of all, the primary cell culture, this is not happened in our India or others any sophisticated laboratory. Basically this type of primary culture, after that the cell line will be produced, then we are using the subculture. So isolation of the cell, how to isolate the cell, this is the steps. Cell are isolated from the any particular tissue, say as for example animal source or the plant source, and disaggregation of the cell. Next, another is the tissue is the aggregated cell, how it will be disaggregated, and this is the steps for the cell are separated from by each other to disrupt their normal relationship of the neighbor cell. And by the basically this disintegration can be achieved by the chemical agents and like enzymes for the trypsin, collagenase and hyaluronidase are the or in the either in the combination. Basically the hyaluronidase, it is the enzymes for the disintegrate the cells, and the sometimes we are using some mechanical by using some tissue homogenizer. This is also another method by which you can separate the cell by the tissue homogenizer by the mechanical method. Fine, then the incubation of the cell in the suitable culture media by the maintenance the optimum culture condition to grow in the isolated cell. That means there are those cells which are isolated from the plant or tissue, then it will disintegrated and it will be placed in the incubator. Then it will be subculturing. Generally, all are the experimental purpose. Generally, we are use the subculture. When the primary culture will be over, then the subculture in everyday. And when the cell of primary culture has grown up and achieved the proper growth state, and there is a subculture to give the more nutritional support for the continuous growth. And the subculturing is necessary steps to maintain the cell in continuous growth phase, because to maintain the growth phase, and after the sometimes the primary cultures of the cell consume the provides nutrients and lack of the nutrient growth because the strengthen and the leads to death. So their media is very important, otherwise this cell will be dead. And thus the maintaining the cell viability, last class we discussed about the cell viability, and the cell are transformed into the fresh culture media. Basically in the subculture, the fresh culture is used, and this fresh culture used for the growth of this compound. And for subculturing, the cell from the primary cell culture are harvested and isolated and transferred to the fresh culture media. And before the subculturing cell can be preserved for the DMSO, dimethyl sulfoxide. Dimethyl sulfoxide, it is the component, or the glycerol by the cryopreservation technique. The cryopreservation is one of the most important technique for the cell animal cell culture. How to be preserved? The isolation of the cell, then the cell will be collected in the tissue or the appropriate pot, then culturing it, then subculture it with the different tubes or in the petri dish, transfer to the culture into the fresh culture media, just like in case of in the microbial cultures, and subculturing and subculturing, and finally the transfer the culture media into the fresh culture media. One or two, so the different types of different numbers of subculture culture and number of cells will be increases. The primary cell cultures, when the cells are surgically removed from the organ or placed into the suitable culture media environment, that will be attached or divided or in the grow. So primary culture is important, but it will be generally occurred by the technical skilled person. After that it will be subculture, and this is the first step of the establishment of the subcultures, and this is called the primary cell culture. This primary cell culture generally not by in the most of the institute, generally it occur in the American Cell Culture Society. And to obtain the primary cell culture, and tissue are cut into the small fragments and enzymatically by using enzyme pepsin collagenase hyaluronidase separate the single cell. And many cell lot of viability, that means lower alive, and but many of them started to grow and divided into the new cells in the after adding a growth culture or the animal culture. If the cell multiply repeatedly and grow for a long time, these cell can also then put into the plastic bottle with a fresh solution. An establishment of the cell culture. When the primary cell culture continue to grow and divided, there the subculture in the fresh media to maintain their viability, growth, and after the subculturing, about 7 times these cells are called the established cell line. That means the primary culture, when it is grow after subculturing by this process, it is grow in the 70 times, that is called the subculture, or sorry, that is called the cell line, and that cell line will be purchased or you can sell it. And the primary cell line can grow very slowly, but established cell line grow faster as there is some alternation in the cell metabolic mechanism due to the many subculture steps. The primary cell maintaining the proper transform the cell with the subsequent subculturing more than 80 times. The establishment cell becomes the immortal, and cell will be the capacity to grow the identified, and normal cell have been the limited growth capacity, but some cell acquire a capacity of growth and finite times, and this capacity is generated due to the transformation in the genetic material, and due to the transformation, cell lose the sensitivity to the external stimuli and also sometimes show the changes in the chromosomal numbers. So transform cell in case of subculturing, why? Then there are the steps, the basic protocol for the primary culture. First, the organ will be collected from the plant or animal tissue. Then the cell fragment will be or tissue. Then the enzyme digested of the cell fragments with the enzymes by the pepsin, collagenase, and the hyaluronidase. A single cell will be occurred, and this single cell will be placed on the suitable culturing media of single cell and the suitable culture media. This is called the primary cell line. If the mixed cell, there is another problem. And subsequent the free sub culturing the around 70 to 80 times the primary culture cell suitable the culture media. This is the established cell culture. In primary cell culture, this is a slow process and the cost effective, but the established cell culture is a very fast process and the less cost effective. After the more than the 70% of the subculturing, there is alternation of the cell division and characteristics. This chromosomal number, cell requirement, indefinite cell growth, that is called the transformed cell culture. There is three cultures: primary, established, and the transformed cell cultures. The types of culture media for the animal tissue culture. This is the another important area. You know it, otherwise you cannot establish the culture. There's various type of culture media used for the culture method and generally categorized into the two broad groups. Number one, the natural media, another artificial media. The media that components of from a natural origin and provide the sufficient nutrient support to proliferation of the animals and the, as for example, clots and the biological fluids, sometimes sera or tissue extract. These are the natural media, or sometimes the human serum also used, the plasma clots, biological fluid, tissue extract, and preparation by the mixing the 15 drops of plasma with the 5 drops of the embryo extract in watch glasses, and the various biological fluid used for the culture media, say for amniotic fluid, pluripotent fluid, and aqueous humor and serum in widely used for to obtain the adult human blood, placental cord blood, and horse or calf blood. They are the biological fluids that are used for the culture media as in natural media. I think this is the better, but this is not. This is the collection is very tough, and there is some ethical problem also. The chick embryo extract is the most common use, but the bovine embryo extract was the used. Others, the are spleen, liver, bone marrow, leukocyte, and extract are the tissue extract for the culture media. So we are discussing about the three types of natural media. Next is the artificial media. They are the chemically synthesized substances. When the natural media is not available, then we can use the some artificial media and the serum containing media or serum free media or chemically defined media or protein free media. So generally artificial media can be classified into the 4 categories, and the synthetic media are prepared artificially by adding several nutrients like the organic or the inorganic components, vitamin, salt, oxygen, carbon dioxide, gases, phase, and the serum protein and the carbohydrate and the cofactor, because the vitamin is essential for the growth of the tissue. Salt also is a very essential component, sodium, potassium, calcium. Oxygen also needed for the cell growth, and carbon dioxide also another, because carbon and dioxide is the carbon source. That's why in case of carbon dioxide, 5% carbon dioxide is also added, and different types of media are used for the composition to definite according to their use. The synthetic media are the 2 types. Synthetic media are the 2 types. Synthetic media also the 2 types, and the serum free media and the serum containing media. That means when the synthetic media, that means which is already discussed, When the synthetic media are devoid of the serum, it's in the culture, it is called the serum free. Only the component are the above component is added to the media, and crude protein fraction such as the bovine serum albumin or the alpha or beta globulin are used for the nutritional supplement, because in case of tissue there are carbohydrate and the protein will be required for the growth and development of the tissue, and they include the serum extract and non-serum tissue extract or tissue hydrolyzed the growth factor hormones carrier protein like the albumin and the transparent lipid metal ions vitamins, which is the animal for this. So serum free media, another is the serum containing media. Serum containing media is a complete of the essential for the growth for the factors hormones attachment of the spreading factors binding and transporting and protein humans bovine serum etc. And serum serve as a source of amino acids protein vitamins, growth factor, lipid, and other nutritional supplement. It also suppresses effects of the shear stress to produce a cell free for physical damage. So these are the discuss about the different media. This is very important for the growth of the media. Therefore, say for example, in case of human beings is the cell culture technique. Another is the cell culture technique. First of the cell culture isolation, then the culture media. Third one is the cell culture technique. Actually, is the dependent cell culture. One of the most first technique for anchor adhere with the and some tissue or cell needed attachment to growth and anchored dependent cell. It is that is called the cell from the kidney. This normal tissue derived cell are the anchored dependent cell and need a surface or any support for their proliferation. Because the proliferation is important, where the uncontrolled proliferation of cell is nothing but the cancer cell, but the proliferation, normal proliferation, it is called the normal cell. This is the normal property of this particular cell. The absence of the any support of surface animal anchored with the dependent cell growth is arrested and cell gets destroyed due to the induction of the propagated programmed cell death, that is called the apoptosis, and these cells are the sensitive to the shear stress, and these cells require to cell communications and interaction and the cell signaling pathway of their survivals, because cell are survive cell growth will be on the signaling systems, and they anchor on the independent cell cultures. The cell which does not require a attachment of the growth do not attach to the surface of the culture vessels and anchor independent cell culture, as for example lymphocytes. The kidney cell are the dependent cell culture anchored, and in the independent cell culture is the lymphocytes, cell derived from the hematopoietic systems and the transplant cell like the cell like cancer cell, myeloma cell, and non-anchored dependent cell. Oh, क्या हुआ? So these are the procedure by which the techniques can use, suppose say for in case of liver cell. First you cut the cell and which is placed on the culture media, and there a cell free culture will be produced in the culture media. So it's a cell free culture will be produced. Next, the one cell will be cultured. You can see it. The normal cell or the stem cells, and the myeloma cell or the cancer cell. Ensure the dependent cell line, another is the ensure independent cell line. The cell proliferation and any support of the surface inside the culture media. That means in that case the supporting media is required, but in case of myeloma or cancer cell, ensure independent or the cancer free cell cultures. The cell does not needed any support surface for the proliferation inside the cell medium, culture media. Here is the medium is required, and here is the support is needed. The application of the cell culture I will discuss in the last class elaborately. Animal cell culture survival for the pharmaceutical industry and the research and production of the valuable biomolecules and the research study for the cell behavior. It is also used in the treatment of the various genetic and the non-genetic causes. And the drug screening development. So drug development and the drug screening, potential use for the therapeutic agent and the screening of the essential and one of the chemical molecules and for the treatment of the disease. It is efficiency for the potency and the checked before the commercial production and cultures are cell exposed in the chemical and tested. So I think it will be better. I already discussed in the previous slides, and the cell-based manufacturing also. Another is the production of the vaccine also. Hepatitis, polio, even the corona vaccine, they are produced by this technique also. Production of vaccine, production of the monoclonal antibody, which is monoclonal antibody much more efficient for the therapeutic purpose. And next is the toxicity study. Last class discussed about the toxicity study. And next is the cancer research. All the cancer research, the culture media, the animal cell culture are transformed into the cancer cell and examined for the therapeutic effect, and any drugs and the chemical agents are either method to help the development of the techniques and the meet the any drug for the cancer treatment. So these are the beyond the virological study we can by or genetic engineering study the cancer cell or the this type of animal cell culture, which is the genetic engineering. So thank you very much for listening the lectures, and especially these are the application of the different types of cell cultures. Thank you very much. Thank you once again.
Sir, wonderful lecture.
Okay.
Yes, sir. Yeah. Yeah, I'll be done.
Good job.
Yeah. It's my boss.
See?
হ্যালো, কিছু বুঝলে? হ্যালো।